Hello everyone. This is a presentation of our work titled The Impact of Missense SNPs on Amyloidogenesis, the example of Αβ and Alzheimer's disease. This work was conducted at the Biophysics and Bioinformatics Laboratory of the National and Kapodistrian University of Athens. Amyloidogenesis is the process by which globular proteins self-assemble into well-defined fibrillar aggregates known as amyloid fibrils. A number of proteins, called amyloidogenic proteins, are capable of going through this process, and many have been associated with a variety of diseases. One such disease is Alzheimer's disease, which is characterized by amyloid plaques formed by the Αβ peptide, a cleavage product of amyloid beta precursor protein APP. Missense single nucleotide polymorphisms, from here on referred to as SNPs, are point mutations that result in a single amino acid substitution and can be characterized according to their role in disease as pathological, benign, or unclassified. In our study, we aim to analyze how SNPs, which occur on amyloidogenic proteins, affect the process of amyloidogenesis. We collected amyloidogenic proteins primarily from AMICO, the amyloidosis collection, but also included data from the latest curated dataset published by the International Society of Amyloidosis. SNPs occurring on those proteins were collected from three databases: DBSNP, CleanVar, and UniProt. Those SNPs were then mapped on the canonical sequence using VarMap. Additionally, the AmyLpred2 algorithm was used in order to predict sequence segments with a high propensity to form amyloid fibrils that likely guide the process of amyloidogenesis. Statistical analysis was performed to determine the significance of our findings, such as chi-squared test and resampling with replacement bootstrap. To illustrate the role SNPs can have on the process of amyloidogenesis and disease, we used APP, Αβ, and AD as an example. The effects for SNPs occurring in APP were extracted from CleanVar, AlzForum, and AMIL. In total, we found over 17,000 unique SNPs on 43 amyloidogenic proteins. We found that the majority of SNPs were actually unclassified. We also see that pathological SNPs appeared at a much higher frequency on predicted amyloidogenic segments, approximately 6% versus 4.4%. Chi-squared tests showed that this difference was statistically significant. The data set of pathological SNPs was very small, so we used resampling with replacement to estimate the statistical significance of all amino acid substitutions caused by pathological and benign SNPs. The most frequent pathological substitution in predicted amyloidogenic segments was that of leucine by proline. This could be related to the destabilizing effect of proline on beta strands, where predicted amyloidogenic segments are often located. Next, we focused on APP and AD. For the production of the Αβ peptide, APP is cleaved by beta and gamma secretase. Gamma secretase has more than one possible cleavage site, resulting in different Αβ species. Post-cleavage, the Αβ peptide may aggregate in order to form amyloid plaques. We found a total of 570 SNPs on APP, 22 of which were pathological. 20 of those were found within or near the Aβ42 sequence. Benign and unclassified SNPs were spread throughout the length of APP. The pathological SNPs affect the process of amyloidogenesis of Αβ in these main ways. They can increase Ab's tendency to form amyloid fibrils. They can also alter the proteolytic cleavage rate of APP, thus resulting in increased production of Αβ peptide. Or they can also shift the analogy of Aβ40 to Aβ40 to favor higher concentrations of the more amyloidogenic Aβ42. The single protective SNP shown in position 2, where alanine is replaced by threonine, reduces APP's affinity to beta secretase, thus lowering the production of Aβ. In conclusion, we found that pathological SNPs are more frequently located in predicted amyloidogenic segments. In AD, such pathological SNPs can increase Aβ production or its propensity to aggregate. Given that APP variants have a significant role in AD, they should be considered when designing pharmaceuticals. This work can be expanded and extended to the remaining 42 amyloidogenic proteins. Feel free to contact us with any questions or comments. Thank you.